Hi class, welcome to chapter 6 on proteins. Today we're going to talk about how proteins are made, their functions, protein metabolism, and then we have a highlight on nutritional genomics. So let's get started. Do you remember the three chemical elements that were found in carbs and fats? Hopefully you said carbon, oxygen, and hydrogen. In proteins, there are four chemical elements that are found. So we have carbon, oxygen, hydrogen, just like fats and carbohydrates, and we have a nitrogen group. So nitrogen is the additional element that is found in all proteins. Proteins are arranged into amino acids, and amino acids are individual units that we're going to talk about coming up, and then multiple amino acids are linked together to create a protein chain, and this protein chain can twist and turn and bond with other protein chains to form shapes that become functional proteins in your body. In addition to nitrogen, some amino acids also have sulfur atoms, and that can give certain protein foods a very distinct smell. So what are these amino acids that I talk about? Amino acids are the building blocks of protein. I think of a protein as like a puzzle, and the end product of a puzzle where maybe you have a mountain scene or a jungle scene or something like that, and each individual piece is necessary to form the big picture. So amino acids are like the individual pieces of a puzzle that don't do too much as single units, but when combined can form a beautiful picture. Proteins have 20 common amino acids, so there's 20 different kind of puzzle pieces in our puzzle, and depending on what the end picture is going to look like, we arrange the puzzle pieces in different ways. So what does an amino acid look like? Every single amino acid has this basic structure. It has an amino group, and the amino group refers to the nitrogen. So the amino group is shown here in light blue. It has a central carbon, and then it has an acid group, which if you remember from our fatty acids, it looks just like this. So it has a carbon with a double bond to oxygen, a single bond to oxygen and hydrogen. So the acid group is shown in the green. And then what makes amino acids different from one another is the side group. So in this particular picture, there's nothing shown in the side group. The side group is just a white box, but the white box could represent a variety of different elements. So for example, in the simplest amino acid, in that white box would be just one hydrogen. In more complex amino acids, we might have multiple carbons or sulfurs or a ring structure. And we're going to see some pictures of different amino acids coming up. So what causes the amino acids to be different from one another is this side group, which in this diagram actually looks more like a top group than a side group, but we refer to it as a side group. In our body, we have some amino acids that are essential, meaning we cannot make them ourselves. And then we have some amino acids that are considered non-essential because our bodies are able to make them. So on the left-hand side of this chart, we have the essential amino acids, and on the right-hand side, we have the non-essential amino acids. And in order for our body to form proteins, we generally need a combination of essential and non-essential amino acids. So if any of these particular amino acids are lacking, especially an essential one, we have difficulty forming protein. If it's a non-essential amino acid lacking, we can synthesize that amino acid ourselves. But our essential amino acids we must get from food sources. This is a picture of all the different types of amino acids. um, and some properties of them. You do not have to memorize this for this class whatsoever, but this just gives you an example of how different they are. Um, in these particular structures, you can see different side groups. Some of them contain pentagon shapes, some of them contain hexagon shapes. These are both considered ring shapes. Some of them contain additional acid groups. Some of them, like arginine, contain additional nitrogen groups. Methionine has a S. S represents sulfur. Cysteine has an S. S represents sulfur, etc. So each amino acid is separate from other ones because of its side chain. So you can see what's similar about them all is the carboxylic acid portion and the NH2, which is the amino portion. And then the side chain is what's different. These are the essential amino acids. So just a second list of them. They haven't changed. They're the same as in the other table, but I'm just putting emphasis on them. So sometimes we call them indispensable amino acids because we can't live without them. And remember, we have to get these from food sources. We cannot make them ourselves. Sometimes an amino acid that is non-essential becomes conditionally essential. And an amino acid that is conditionally essential means that under certain conditions, we're not able to make it ourselves. So one example is tyrosine. Normally, the body can make the amino acid tyrosine, which is not considered essential, from phenylalanine. In certain cases of disease, there's a particular disease called phenylketonuria. People are not able to convert phenylalanine to tyrosine. And so tyrosine actually becomes conditionally essential, and they have to get tyrosine from food products. On the flip side of that, since they're unable to convert phenylalanine to tyrosine, the phenylalanine builds up to high levels, and this can actually become toxic for them. So this disease, like I said, was called phenylketonuria, or PKU, and it's a very rare genetic condition. I actually met one patient with this at Cal Poly. She came to my class and spoke about the different dietary habits she had to have because of this disease. These are more pictures of amino acids, and in the white box you can see the different side chains of them. So glycine is one of the simplest amino acids. It just has a hydrogen side chain. Alanine has a methyl group, which is a carbon and 3 hydrogens. Aspartic acid, when you see the name acid, it indicates carboxylic acid. So you can see this particular amino acid has an additional acid group in its side chain. And then phenylalanine has a carbon ring, which is a 6-carbon ring. And the structure of each of these amino acids gives them unique properties so that they can form different proteins with different functions in the body. Peptide bond. So how do these amino acids connect with one another? How do we put our puzzle pieces together? So we connect our puzzle pieces via something called a peptide bond. And a peptide bond will connect the acidic end of one amino acid with the amino end of another. And then what happens is hydrogen and oxygen can be lost. So depending on how many peptide bonds are formed, we have different length proteins. We have similar terminology to what we had in our previous lectures with fatty acids. And so we have di, which means 2, tri, which means 3, and poly, which means many. So in a polypeptide, we have more than 3 amino acids linked together. In a dipeptide, we have 2 amino acids linked together, and a tripeptide, we have 3 amino acids linked together. So I kind of hinted to this reaction, but if you guys remember the reaction that can bring two monosaccharides or sugars together, it's actually the same reaction that can bring two amino acids together. So when we bring something together, water is lost and we have a condensation reaction. This is a picture of the condensation reaction when you form a peptide bond. So in all condensation reactions, H2O or water is lost. And you can see in this particular example, an oxygen and a hydrogen group are lost from the amino end of an amino acid, and a hydrogen is lost from the an oxygen and hydrogen group is lost from the acid end of an amino acid, and a hydrogen is lost from the amino end of an amino acid. And then that acidic group, the carbon with the double bond to oxygen, instead of having the OH, that's replaced by nitrogen. So that carbon will be connected directly to nitrogen to form a peptide bond and a dipeptide. And you can have proteins that are many, many amino acids long and have multiple peptide bonds. What roles do proteins play in the body? We're going to talk about many of the roles coming up, but they are involved in our immune system, they're involved in our muscles, they're involved in vision, they're involved in acid-base balance, they're involved in healing from wounds, so many things. They produce hormones, they're involved in enzymes, just so many vital functions of proteins in the body. What allows proteins to play different roles in the body is protein shape. And protein shape can be described in four different kind of categories or structures. We have primary protein structure, which is just the sequence of amino acids. We have secondary protein structure, which involves polypeptide shapes that are created between different sequences of amino acids and weak electrical interactions. We have tertiary structure, which is more like, we call it a polypeptide tangle, but it's not a tangle in the sense that it's chaotic. It's a very organized, folded form. And then we have quaternary structure, which will involve interactions between two or more polypeptides. And we're going to see some examples of this coming up. So, some of the functions of protein based on their structure. Insulin. Insulin, remember, is a hormone, and insulin helps bring glucose into the cells. Insulin is simply a protein. You have two long polypeptide chains, and they're shown by the pink above. In each pink circle, you have a three-letter abbreviation, and those three-letter abbreviations each stand for a separate amino acid. So, for example, ALA is alanine, VAL is valine, LEU is leucine, HIS is histidine, etc. So you have a kind of this top protein chain, and then you have this bottom protein chain, and in between the two polypeptide chains are S's, and S's represent sulfur. So these are connected by what we call sulfur bridges. And by connecting two polypeptide chains this way, we form the hormone insulin. So insulin is actually a very simple protein. Hemoglobin is a protein. Hemoglobin is a much more complex protein, and hemoglobin actually represents quaternary structure, which is the most complex protein structure. Hemoglobin is composed of four different polypeptide chains. Each chain, which is represented here by the colors yellow, blue, green, and purple, has its own shape. So each of these chains has its own shape. And the shape of each chain allows an iron and a heme molecule to sit within that particular shape. And then within the hemoglobin molecule itself, there are four shapes that are bond with weak chemical connections. Protein denaturation. So, protein denaturation involves changing the shape of a protein. And when we change the shape of a protein, such as that hemoglobin or the insulin, the protein doesn't function the way it was originally intended to do. Some things that affect proteins and can denature them or change their shape include heat, so such as cooking, agitation, such as whipping, acids, maybe something like lemon juice, bases, alcohol, heavy metals, which hopefully we're not exposed to, and many other agents. So, an example of changing a protein shape would be when you cook an egg. The egg starts out very liquidy, it's clear, the yolk is runny, and then when you cook it, the clear part, the white, becomes white versus clear, the yolk becomes harder, and that protein shape is forever changed. It's no longer an egg capable of, producing a baby bird. Other examples, again with an egg, would be whipping egg whites to form peaks. So maybe this is done when you're making like meringue for a pie. Once you whip the egg whites so much and making the meringue peaks, you can't reverse that process. Also, things like the whipping of cream to turn into butter, or the whipping of cream to turn into whipped cream. That changes the protein shape of those molecules. In our stomach, we have hydrochloric acid, and this can also denature proteins so that we can break them down. Hydrochloric acid also activates something called pepsin, which we're going to see coming up on our next slide. Pepsin is an enzyme that can cleave or break proteins apart. So pepsin can take long polypeptide chains and break them apart into smaller polypeptides, and in some cases, single amino acids. So this is an overview of protein digestion. In our mouth, we start by chewing our foods, and this can break down the proteins into smaller particles so that they'll be exposed to more enzymes. It will increase the surface area of the proteins. But we really don't have a lot of digestion or absorption happening in the mouth. It's just this kind of mechanical breakdown. Once proteins hit the stomach, they're exposed to our stomach acid, hydrochloric acid, and hydrochloric acid can denature the protein shape. Hydrochloric acid also activates an enzyme called pepsinogen, which is kind of like a precursor to an enzyme to pepsin, which is an active enzyme. And I already discussed that pepsin can break proteins down. Once proteins move out of the stomach into the small intestine, we have pancreatic juices that are going to be released. We have proteases that are going to be released, and those can further break down polypeptides into smaller proteins. We also have enzymes on the surface of our small intestinal cells that can further break the peptides down, which are protein chains, into single amino acids. And you can see in this light purple column different enzymes that can contribute to protein breakdown on the right-hand side. Proteins in our body. So the human body contains genes, and genes are our genetic material or our blueprint that codes for thousands and thousands of proteins. So genes, or our genetic material, is DNA. DNA and RNA can code for proteins in a process called translation. Protein synthesis can occur. So for this class, you do not have to memorize the details of protein synthesis, but I'll show you a diagram on the next page, just kind of an FYI. But you should know that our genetic code, or our DNA, is involved in protein coding, protein synthesis, and translation. So this is a rather complex drawing of protein synthesis. And DNA serves as a template to make something called messenger RNA. And each messenger RNA copies DNA exactly, so it has the same kind of genetic code. mRNA leaves the nucleus of the cell, and DNA stays inside the cell. mRNA will attach to a protein, making something called a ribosome, or will attach itself to a protein-making machinery called a ribosome, and the ribosome will subsequently dictate the ribosome will help form this amino acid chain by adding one single amino acid at a time and moving down the mRNA, which the mRNA serves as kind of a blueprint. So as the ribosome moves down the mRNA and single amino acids are added at a time, eventually the amino acid chain becomes a lot longer. So what would happen if we're making these proteins and instead of the correct sequence of amino acids, we got the wrong sequence of amino acids? So this would be like if you had two puzzles and some of the pieces from the first puzzle got put in the box with the second puzzle. What would happen if we had the amino acid sequence differently than what was originally coded in the DNA? If we get the amino acid sequence incorrect, it can significantly change the shape and the function of the proteins. An example here is sickle cell anemia. So remember that hemoglobin is a protein, and hemoglobin is a component of our red blood cells, which are shown in this diagram. This circular red object is a red blood cell, and normally it's kind of concave, doesn't have a nucleus, it's disc-shaped, and this allows it to move very easily through the bloodstream as well as carry oxygen. Normal hemoglobin is coded by valine, histidine, leucine, threonine, proline, and I believe this is glutamate or glutamine. However, in sickle cell anemia, this is glutamic acid, sorry. In sickle cell anemia, instead of one of these glutamic acids, you have a valine instead. So it's like you have this wrong puzzle piece being inserted into your puzzle, and because of that, the puzzle doesn't look the way it's supposed to. The hemoglobin, remember, which was made out of 4 different protein chains, and they were all folded together, the hemoglobin no longer has that organized shape, and instead it forms this sickle shape. And the sickle shape actually can get stuck within the blood vessels, it can cause interruptions within the vascular system, and it doesn't carry oxygen very effectively. So therefore, people with sickle cell anemia have shortened lifespans and multiple complications due to the shape of their hemoglobin. Roles of protein in the body. So I already discussed a few of these roles earlier, and now we're going to talk in a little bit more detail about some of them. Proteins can serve as building materials. So remember, proteins are our puzzle pieces or our Legos to our Lego kit. They're literally the building blocks of every single protein that we make. One example is collagen, and collagen is a structural protein from which connective tissues, such as our scars, our tendons, our ligaments, bones, and teeth are made. Protein helps form a matrix, which is, in this particular example, a bony matrix. And the matrix is kind of the basic substance that gives the form to the developing structure in the body. So proteins are important components of all of our structures in the body. Enzymes are also proteins, and enzymes are needed to advance chemical reactions and facilitate chemical reactions. Enzymes have a very distinct shape so that they can bind what's called their substrate, which in this particular picture is circular A molecule and B molecule. And you can see that this enzyme shape has like two half circles in it so that the A and the B will fit perfectly. But there are many, many different enzymes in our body, and they all have different shapes, and their shapes are determined by the single amino acid sequence and the way that those polypeptide chains twist and form within one another. Proteins function as hormones. I already mentioned insulin. The counterpart to insulin is glucagon. We also have growth hormone. We have thyroxine, which is one of our thyroid hormones. We have calcitonin and parathyroid hormone. These are involved in calcium balance and bone calcium balance. And we have antidiuretic hormone, which this has actually been changed, but this regulates fluid balance. The name of antidiuretic hormone has been changed, not what it actually does. Proteins affect fluid balance. So within our cells, we have proteins, and proteins attract water. So when we have adequate amounts of proteins inside our cells, there is adequate amounts of water inside our cells. When we have protein that leaks out of our cells, or we don't have enough protein, we're unable to bind water, and water can fill these intracellular spaces. And so when we have water leaking outside of the cells, filling these intracellular spaces, something called edema develops. And edema is the presence of fluid or water outside of the cell between these intracellular spaces, and outside of the blood vessels as well. So in this particular picture, they're showing somebody's ankles, which is a very common spot for edema development. And they have pressed their finger into one of the ankles. And then in the picture on the right, that finger stays indented. So this is representing fluid that has moved outside of the cells, due to likely protein losses, inadequate protein synthesis, inadequate dietary intake, or there's a couple other things that can cause edema as well, but edema can be protein related. Acid-base regulation. So proteins have negative charges on them, and because of this negative charge, they're able to attract hydrogen ions. Hydrogen ions have positive charges. And so by attracting hydrogen ions and then sometimes releasing hydrogen ions, they can contribute to acid-base balance in our body. It's very important that our blood is maintained at a specific pH. I believe it's usually between 7.35 and 7.45, and proteins can help maintain that pH by kind of juggling hydrogen ions. If the pH becomes too low, something called acidosis occurs. And so acidosis would be when the blood has a very low pH or an acidic pH. And if the pH becomes too high, alkalosis occurs, meaning a high pH or basic pH. And at either too high or too low of a pH, proteins can be denatured. And remember, we've just said proteins form enzymes, proteins form hormones, proteins form building blocks. And so if proteins are denatured, all of the functions of proteins can be affected. So it's very important that we maintain a stable pH within our body. Proteins can serve as transport molecules. So, for example, proteins can bridge the gap between the inside of a cell and the outside of a cell by forming within cell membranes. For example, there is a protein called the GLUT protein, GLUT, and the GLUT protein moves to the cell wall and allows glucose to come into the cell wall in the presence of insulin. Other examples of transporting proteins are hemoglobin, which transports oxygen, and lipoproteins, which hopefully you remember from our last chapter. Remember, lipoproteins are like our fat taxis, transporting cholesterol, triglycerides, and proteins around our body. Proteins are important to our immune system. So anytime we are exposed to a potential pathogen, we can form antibodies to that pathogen. And antibodies are immune components that help us fight off disease. Antibodies are made from proteins, and proteins give antibodies a specific shape so that we have different antibodies for each antigen. For example, we may have antibodies to certain strains of a cold virus. When we get vaccine, we can form antibodies to potentially hepatitis or chickenpox or pneumonia. And the different protein structures, the sequence of amino acids within those antibodies, gives them a distinct shape so that they're not all identical. In cases of protein deficiency, we don't have enough protein available to form these antibodies. So in cases of protein deficiency or severe malnutrition, our immune systems end up weaker. We simply don't have the building blocks to fight off disease, and so we're at higher risk for contracting and becoming sicker from disease. We do not like to use protein as energy. Our body would much rather use it for all the functions I previously mentioned. However, in cases of starvation or severe calorie malnutrition, we can use protein for energy. And protein yields 4 calories per gram. The process that can make glucose from non-carb sources such as proteins or amino acids is called gluconeogenesis. And we're actually going to jump into gluconeogenesis with our next chapter, metabolism. But again, like I said, our body does not like to use proteins as fuel. We'd rather use carbohydrates and we'd rather use fats. Additional roles in proteins. Proteins form important structures in our eyes. They can form structures called opsins, and opsins allow us to respond to the presence of light. They're light-sensitive pigments, and those light-sensitive pigments send nerve impulses that allow us to see visual images. Protein metabolism, I'm just going to touch on this basically because, like I said, we're going to be covering this a little bit more in our next chapter, but I'll just touch on it here. So protein turnover. Protein turnover is kind of like recycling or rearranging. And so protein turnover involves breaking down proteins or degradation and then building new proteins. When we break down a protein along, so say like a long polypeptide chain or a hemoglobin molecule, we have all of those individual amino acids that we can now use to build something else. And so this is actually a constant process in our body where we're breaking down proteins, we're reusing those amino acids to make something else. When we break down proteins, the amino acids go to what's called an amino acid pool, and it's not a literal swimming pool, but it's a storage area for free amino acids from which our body can draw from. So because we actually have an amino acid pool, if we're eating foods that don't necessarily have all of the amino acids we need, on a short-term basis, we can pull from that amino acid pool to get the complementary amino acids. If we're chronically eating foods that don't have all the amino acids we need, eventually that amino acid pool will become depleted of some of those essential amino acids, and we won't be able to draw on it from it anymore. But on a short-term basis, the amino acid pool allows us to eat a variety of foods with different amino acid profiles, and we have kind of a little bit of reserve within our body. Nitrogen balance. So you remember that every single amino acid has the amine group, and the amine group has nitrogen in it. So when we eat protein foods, we are, in one sense, consuming nitrogen. We're intaking nitrogen into our bodies. Nitrogen can be involved in forming some toxic compounds such as ammonia, not pneumonia, ammonia. So ammonia with an A. And so our body generally will get rid of nitrogen. We can also use nitrogen as a building block. So in some cases, we hold on to nitrogen. And the balance between the nitrogen we put in our bodies versus the nitrogen that we excrete is considered nitrogen balance. In healthy, full-grown adults, we have equilibrium, meaning the amount of nitrogen we take in is equal to the nitrogen we take out. When children and infants are growing, they take in more nitrogen than they put out. And when I say put out, I mean excrete via urination. And the reason that children retain more nitrogen is because they're using this as building blocks for new proteins, because they're growing muscle, they're growing new cells, they're growing taller, they're growing bone, they're growing new blood supply, etc. In cases of negative nitrogen balance, we're not taking in as much nitrogen as we're excreting. So this can happen in starvation, where we simply don't have enough nitrogen. This can happen in severe injury and also in infection. Using amino acids to make fat. So remember on our last lecture, we talked about fats contributing to weight gain. And what I said was that any component in excess can contribute to weight gain. And that's because any dietary component, any of the macronutrients, carbs, protein, or fat, if eaten in excess, can be used to make fat. And so that can then contribute to weight gain. So if we're exceeding our protein intakes and we do not need any more, we can take those proteins and convert them into fats. So it is possible that somebody following a high protein diet can have fat production. How do we get rid of proteins in the body? So I just talked about nitrogen balance, and I talked about nitrogen output versus nitrogen input. We get rid of proteins at the end of it all via the kidneys, but proteins, specifically amino acids, have to go through multiple steps before the kidneys can get rid of them. So what happens is something called deamination, and deamination is removing the amino group from those proteins. So in the liver, the amino group is removed, and that amino group combines with hydrogen and produces something called ammonia. Ammonia is toxic. You may have seen it in household cleaning products. So we do not want ammonia to build up in the body. In order to prevent ammonia from building up, the liver will combine the ammonia with carbon dioxide, or CO2, and something called urea will be produced. And urea is a lot less toxic than ammonia. Urea will then go into the bloodstream, and remember, all the blood is filtered by the kidneys, so the urea-containing blood will be filtered by the kidneys, and the kidneys will be able to excrete urea in the urine, and that's how we are able to get rid of these nitrogen groups. Protein quality. So what makes a protein higher in quality than another protein? There's a couple different kind of ranking systems or components to consider when discussing protein quality. And those include digestibility, how we can break down these proteins. Animal proteins tend to be about 90 to 99% digestible. Plant proteins tend to be 70 to 90% digestible due to high fiber components. Are we eating other foods at the same time that we're digesting proteins? Do we have adequate stomach acid, etc.? Amino acid composition. Does the protein have all of the essential amino acids in the right amounts? A reference protein. A reference protein is something that's used as a comparison to kind of rank different proteins. So a reference protein will provide all the amino acids needed for growth of a preschool age children. And so when we compare things to reference proteins, such as eggs, we can see how closely they rank to this. We have high quality proteins. These are going to contain all the essential amino acids in the proportion we need. And then we have complementary proteins, which we'll discuss. So quality. Protein quality determines how well children will grow, as well as how well adults will maintain their health. So our protein quality can influence all the functions that protein plays in the body, such as hormone, enzyme production, matrix, bone production, teeth production, muscle, immune system, fluid balance, acid-base balance, etc. And so certain things influence protein quality. Can we digest the proteins, and what is their amino acid composition? So once we do digest them, what do we break them down into? Protein digestibility has to do with how many amino acids we absorb from given protein. So it can vary depending on the source of the protein or other foods eaten with it. Like I said, animal proteins tend to be more digestible than plant proteins. Plant proteins can sometimes be trapped by different fibers within the plant, or potentially plant proteins may be trapped within like an insoluble seed, and if we don't chew that seed, we don't access the protein within the seed. So eggs are pictured here because they're a very digestible source of protein. Reference protein. So reference protein is a standard to which we measure the quality of all other proteins, and they're given a value or ranking. So egg is digestible as well as it contains amino acids that we need. So it's given a reference value of 100, and then we can rank other proteins compared to eggs. So we could rank wheat compared to eggs. We could rank soy compared to eggs. We could rank whey protein compared to eggs. We could rank quinoa compared to eggs, etc. High-quality proteins contain all the essential amino acids in the amounts that humans need. And so when we get all the essential amino acids in the amount we need, we have all the building blocks so that we can reassemble proteins into structures within our body. Limiting amino acids. So some protein sources contain most of the essential amino acids, but may be low in some of them. And when they're low in a few of the essential amino acids, those essential amino acids are considered to be limiting factors for protein synthesis, meaning if you didn't have enough of them, you wouldn't be able to synthesize proteins in your body. So because certain foods do have limiting amino acids which they're not necessarily rich in, we can consume foods that are considered complementary. So foods that are complementary complement each other by having different amino acid profiles. And I'm going to show some examples on the next page coming up. But here pictured is kind of a veggie bowl. And it's got some grilled tofu. It has a little bit of non-dairy. It has corn. It has onions. It has red beans. It has peppers. It has a little bit of cheese, rice. And so while there's not necessarily an animal protein in this bowl, because of all the different plant proteins, all the essential amino acids will be found within this meal. So, examples of proteins that have limiting amino acids. Legumes, such as beans, pictured here, have low levels of methionine, which is MET, that's why it's blank white box, and tryptophan. Methionine and tryptophan are both essential amino acids. So legumes have limiting amounts of methionine and tryptophan. Grains, on the other hand, have limiting amounts of ILE, which is isoleucine, and LYS, which is lysine, both also essential amino acids. However, when you eat grains and beans together, you're going to get the essential amino acids that the other product was missing. So when you eat grains with legumes, the grains are going to compensate for the low methionine and tryptophan in the legumes. When you eat legumes with grains, the legumes are going to compensate from the low isoleucine and lysine in the grains. They used to think that you had to eat this at the exact same meal to get all the amino acids you need, but now they've learned that it's okay to eat these foods throughout the day because of our amino acid pools. So if you ate legumes in the morning, you would have high levels of isoleucine and lysine. Those could be moved to your amino acid pool as kind of a reserve. And then, say later in the day when you ate grains, you would be able to draw from that amino acid pool for isoleucine and lysine, and you would deposit into that amino acid pool methionine and tryptophan. Health effects and recommended intakes of protein. So in these two pictures, you see two cases of what we consider protein energy malnutrition. And on the left, there is a child who you can see his rib cage. He's very skinny. This child is suffering from protein energy malnutrition, where this child is not getting enough protein or calories. And this child has something called marasmus. The child on the right, he is also pretty skinny, but you can see his belly is protruding, and he's not quite as skinny as the child on the left. So the child on the right is suffering from protein malnutrition, but not necessarily calorie malnutrition. So the child on the right is probably eating food, but eating starchy foods like rice or cassava or taro or plantains, but not getting enough protein. And I know he's not getting enough protein because you see this edema, which in his particular case is occurring in his belly. So he has this protruding edema in his belly. So protein energy malnutrition or protein calorie malnutrition can be categorized into two different types, which I was describing based on those pictures. And the first one is marasmus, where you have inadequate calories and protein. And then the next one is kwashiorkor, where you have inadequate protein, but you do have some level of calories. Health effects of protein. So protein plays a role in disease, and it's really kind of about finding the balance as well as eating healthy types of proteins. We obviously need adequate protein for heart function, but too many animal proteins have actually been associated with heart disease, where plant-based proteins seem to be protective of heart disease. Some similar findings go to cancer. Certain intakes of animal proteins, especially high processed red meats, are linked to colon cancer, where plant-based proteins generally are not. Osteoporosis. If we do not have enough protein, we can develop osteoporosis because, remember, protein can help form the bone. However, very, very, very high intakes of protein actually can lead to higher levels of calcium excretion in the urine. And losing calcium in the urine may potentially play a role in the development of osteoporosis. So it's kind of about finding the happy medium with protein intake. Weight control, like I said, too much of anything can contribute to weight gain. And then kidney disease. The protein or the kidneys are needed to break down and excrete urea. And so in cases of chronic, very, very, very high protein intakes, or when the kidneys aren't functioning adequately, protein can actually be harmful to the kidneys. So very, very, very high chronic intakes of protein or just high intakes of protein in the face of kidney disease can make the kidneys work harder. Recommended intakes of protein. So the RDA for protein is individualized based on body weight, and it's currently set at 0.8 grams per kilo of healthy body weight. Certain people may need more protein than this, certain people may need less, but in general, 0.8 is our baseline. Athletes may need between like 1.2 to even 2 grams of protein per kilogram per day, depending on their athletic abilities and athletic training schedule, athletic goals, etc. How to calculate recommended protein intakes. So you would start with a weight. So in this particular example, I have 150 pounds. You don't have to look up a healthy weight for a person your height. You can look up your own weight. So you can know your own weight. So you're going to take your weight and then you're going to convert pounds. You're going to convert pounds to kilograms. So in this particular example, we're going to have 150 pounds and we're going to divide this by 2.2 because there are 2.2 pounds per kilogram. And what we're going to get is that a 150-pound person weighs 68 kilograms. And then we can take this 68 kilograms and we can multiply by the RDA for protein, which is 0.8 grams per kilogram. And what we're going to get is 54 grams of protein per day. So that would be the recommended amount based on the RDA for a person who weighs 150 pounds. So you should be familiar with how to determine how much protein somebody needs, and I think you'll have a couple examples on your study guide. Okay, now I am stuck in drawing mode. Okay, cleared my drawing. And, sorry, I'm stuck in this drawing mode. Okay, so MyPlate recommendations for protein. You can go to myplate.gov and see different recommendations for protein. It will tell you exactly how many servings of protein there are in different foods, different qualities of protein, what's recommended per day based on your calorie level, etc. So MyPlate is a really good source for protein. How much protein is in the average 2,000 calorie per day diet? So this would be an example. If you're consuming 6 ounces of grains, which would be like 6 pieces of bread or maybe 2 large tortillas, 6 small tortillas. In general, grain-containing foods have about 2 to 3 grams of protein each. Vegetables tend to have about 5 grams of protein per serving. This is a little bit less than that. Milk tends to have about 8 grams of protein per serving. And then meat or meat alternate, which would be like tofu or textured vegetable protein, etc., tend to have about 7 or a little bit higher grams of protein per serving. So you can see during this day, this person did consume meat and dairy, which are maybe obvious sources of protein, but they also got a significant amount of protein from their vegetables and grains. Protein and amino acid supplements. So are supplements necessary? I think supplements do have a place, but I do not think everybody needs supplements. Some supplements can be dangerous if they contain single amino acids. If we get too much of a single amino acid, it can be harmful to our body, and our bodies were not meant to consume just singular amino acids in high amounts. Our bodies were meant to consume a variety of amino acids at one time. So certain supplements that are only high in maybe one particular amino acid could potentially be harmful. Many people might take a supplement that has branched chain amino acids, which is going to be a set of 3 amino acids. For the most part, in moderate amounts, those don't tend to be harmful in healthy adults. If you have kidney or liver disease, that's another story. I think people who are not getting enough protein from food sources, those types of people might benefit from supplements. But I don't think that everybody needs them. So I think that they have a place, but they're not necessary. I think they can be used even daily in moderation, but you don't need them. It's very possible to get all the protein you need, all the amino acids you need from food sources alone. Nutritional genomics. So nutritional genomics is kind of the intersection between the human genome and the foods that we eat. And it's really interesting because the foods that we eat can actually change our genetic expression, and our genes can actually cause some alterations with the foods, breaking down, metabolizing, absorbing, excreting the foods that we eat. So they have this kind of two-way relationship. So nutrigenomics is the study of how nutrients affect genes, and nutrigenetics is the study of how genes affect nutrients. And this is a really evolving area of research. We're learning more and more about this every day, especially with the accessibility of our human genome now. It's possible to get a breakdown of your human genome for relatively inexpensively, where this used to cost thousands and thousands of dollars. So even programs out there, which I'm not supporting this one necessarily, but things such as 23andMe, you can analyze your genome, and then if you download the raw data report from 23andMe, you can put it into other databases. There's one called Prometheus, and it will give you your exact genetic breakdown. And then you can actually put that through searches for food-related genetic issues. So for example, I've analyzed my genome through 23andMe. I took the raw data, I ran it through Prometheus, and then I took that data and ran it through a few nutritional search programs, and I learned that I have a deficiency in one of my enzymes that is used to metabolize folate. I have a deficiency in a vitamin D receptor, so I may need higher levels of vitamin D than other people. And I have a susceptibility to high vitamin A levels. So you can learn different things about your genome based on your DNA and the interactions with food. The idea with this is in the future, we're going to be able to do customized diets based on people's individual genetic code. So we could find a diet that's perfect for them. This will also help with disease prevention. So I mentioned PKU, or phenylketonuria, which is the disease where we cannot convert phenylalanine to tyrosine. So we may be able to use nutritional genomics to help with some of that process. This may help for people with high levels of blood cholesterol or altered fat metabolism. All right, the quiz. So, which part of its chemical structure differentiates one amino acid from another? Hopefully you said its side group. Isoleucine, leucine, and lysine are all C, essential amino acids. In the stomach, hydrochloric acid A, denatures proteins and activates pepsin. Proteins that facilitate chemical reactions are called B, enzymes. If an essential amino acid that is needed to make a protein is unavailable, the cells must. How are the cells going to get it? What they're going to do is they're going to break down another protein to obtain it. Protein turnover describes the amount of protein synthesized and degraded. Which of the following food proteins has the best assortment of essential amino acids for the human body? It's egg. Which of the following is a consequence of excess protein intake? This should be C. The more protein you intake, the more nitrogen you break down, and then you have to get rid of more urea. The Protein RDA for a healthy adult who weighs 180 pounds is, it is B. So with this one, remember you're going to have to convert 180 pounds to kilograms, and then you're going to multiply the kilograms by 0.8 grams per day. Which of these food has the least protein per half cup? So I didn't mention this yet, but you may know, but in general, fruits have the lowest protein of all of the food groups. So the answer is orange juice. All right, thank you guys for listening, and I will see you next time.